Okay, so we're going to go ahead and get started. Thank you, everybody, for joining. My name is Chris Benko, and I am the CEO and one of the co-founders of Koneksa Health. Um, Today, primarily, you're going to be hearing from my friend, co-founder, and colleague, Dr. Rob Ellis. I'd like to take a minute just to tell a brief introductory story about some of the origins of Koneksa and how Rob and I got connected, and why it matters to the content at hand today around implementing digital endpoints in CNS trials specifically. I had the opportunity to begin working on Koneksa as an entrepreneur in residence for Merck's Global Health Innovation Fund, which is a strategic venture capital fund that's part of the US organization Merck, known as MSD outside the US. And they helped provide incubation support for the beginning stages of the company, going back to as early as 2014. And in the first couple of months of exploration, we had this loose concept that there was significant untapped potential from the emerging technologies like wearable devices, mobile apps, and sensors that could be applied in a healthcare context and whose data could contribute in meaningful ways to healthcare advances. And as we were starting to try to develop our focus, we had conversations with a lot of different informative stakeholders, one of whom was incredibly impactful and important to us was Gary Herman, who at the time was the head of early development at Merck, and he's currently the senior vice president of early development at Regeneron, and has been a great friend and collaborator throughout Koneksa's journey. And I sat down with Gary in probably March of 2014, and I was explaining to him some of our ideas, and he said, Chris, referencing the program Perlatolant, which had been a compound that came in through the merger with Schering-Plough and was targeted at Parkinson's disease. And he said, Chris, based on the data that we've seen for Perlatolant, and Gary's job as an early development head was to be a skeptic, but he said, I really believe that program should have worked. However, in our proof of concept study, the phase 2B study that put the end to the program, the primary endpoint was a paper symptom diary. And he looked at me and said, now come on, how can we expect a 70-something-year-old patient with Parkinson's to recall effectively what their symptoms were for the last week? I don't think you and I could recall what we had for breakfast 2 days ago. And don't you think something like a device or a sensor or the technologies that you're working with could do a better job of helping us understand their symptoms? That conversation really stuck with me. And it ended up having a significant impact on our decision to focus specifically on developing digital biomarkers to help novel drug development. Perhaps 6 or 8 weeks later, I had the first opportunity to meet Rob at a WeWork facility in Lower Manhattan. And I shared that same anecdote with him as we were still in very early stages of conceptualizing what this company might be. Rob originally trained as a geophysicist. His PhD from Leeds is actually in numerical modeling, but I don't think he's done any geophysics work since leaving university. He spent most of his professional career in industry and financial services, developing applications that utilize big data in the financial sector, before leaving that segment to spend a number of years working on startups, both around education and financial services. And Rob shared with me after hearing that anecdote that his dad was suffering from Parkinson's in the relatively late stages of the disease. And that factor had a specific motivation and impact to him in wanting to consider where he could apply his skills to really make a difference in healthcare. So our initial connection was really all about helping people with diseases like Parkinson's and how much that mattered to both of us and to him, especially from a personal perspective. Unfortunately, while we would lose Rob's dad, no less than, I believe, about 2 years later, perhaps 3, we did have an opportunity to include him as one of our earliest product testers, applying a Fitbit to him to learn things about his activity and sleep patterns that I don't think anyone, including Rob's mom, would have otherwise been able to ascertain. And it was one of our sort of earliest N-of-1 experiments that showed us the power and potential of devices to provide measurements and understanding of disease that we wouldn't otherwise be able to get just by interviewing a patient or the people around them. One of our earliest studies in the pharmaceutical industry, going back to 2016, was in a movement disorder. We had the opportunity to work with Ultragenyx Pharmaceuticals, which is a phenomenal rare disease company, on an ultra rare condition known as GLUT1 deficiency syndrome, which is primarily a movement disorder, although it's characterized by a wide range of symptoms, some of which can be quite debilitating to the children and adult patients that suffer from this disease. And it was a phenomenal experience that developed a great natural history study, and where we had the opportunity to work on their phase 3 to look at how things like activity measures could be used to effectively characterize disease. It wouldn't actually be until a few years later, perhaps the 2018, that we had the opportunity to jump into Parkinson's, although we've worked across a range of neurological conditions, primarily neurodegenerative disorders. And as Rob will explain, we've had the opportunity to build on the great work of a lot of other organizations that have spent time in this space. So with that, I'd like to turn it over to Rob to take you through some of the more technical and scientific aspects of that journey, and hopefully shed some light on the potential for digital endpoints to really be transformative in CNS drug development. So Rob, over to you.
Thank you, everyone. Adam, if we could have the next slide, please. So to some extent, this slide could be descriptive of the digital biomarker challenge overall, certainly in terms of the opportunities and promise that people hope for from digital biomarkers. The challenges on the right-hand side are to some extent specific to CNS conditions. I think in a post-pandemic environment is acting as an accelerator for the deployment of at-home measures in general. but really, the underlying scientific or numerical reasons for doing this are predominantly so you can capture lots of fine-grained objective measures. Lots of measures should really mean a reduction in noise, or more specifically, the ability to isolate individual symptoms and to be able to quantify the severity of those symptoms in ways that a questionnaire might not capture. The challenges in CNS space are broad, but I pick a few of them here. In many CNS disorders, multiple sclerosis, PD, dementia, the gold standards are quite large composite scales which cover a number of different domains, typically require trained raters to administer, and they're not really readily suited for a patient to routinely complete at home. So what the end state objective here is to be able to independently and separately capture both the symptoms that are recorded in those questionnaires through things like device sensors at a point in time or 24/7, to understand the impact of symptoms on the activities of daily living. One of the challenges that we see in some CNS disorders is that a patient's symptoms can vary over time. Not every Parkinson's patient will start out with tremor. Gait and balance issues tend to come later. So when you're trying to study a disease like this, you kind of have to have a very wide field approach and capture as many of the symptoms of interest as possible, but then also solve the problem of figuring out how do you combine them, how do you think about those individual measures in terms of an overall score, because at some point, if you're to use these objective measures as an endpoint, you're going to have to track back to the current gold standard. The challenge here is that sensor data from any number of devices, an actigraph, an iPhone, whatever it might be, they record lots and lots of data. And the hard part is being able to pick out from that data the signal which is really related to the symptom itself. Next slide, please. So this is a general journey. In this case, we have a look at Parkinson's disease, but There are many different flavors of composite scale used in rare diseases now too, like pecan or Batten's disease and so on. On the left, you see some screenshots of different questions from the UPGRS, where in each case, a symptom is being assessed by a clinician and then being scored in severity from 0 to 4. This is a categoric scale. It's an interpretation made by the trained clinician as to the severity of the symptoms that they're being observed. On the right-hand side, you have a digital solution, the solution we have today. And the bottom there shows the journey that you need to go through, and we'll dip into some of those aspects of that journey later. What you see in this example on the right-hand side is a set of tasks that the patient is going to perform at home. And in this case, that the top task, speech, is highlighted. That's the task that this particular sponsor cares about the most. So you will want to put that task at the top of the list to make sure that you record that data, even if you record not much of anything else. Next slide, please. So as Chris mentioned, we make no claim to be the first to do this work, and in actual fact, I think in some cases it's often only because this work is available in the published literature that there are solid foundations to be built upon that enable you to build a clinical grade tool. When I say clinical grade, I don't mean registered as such, but one which demonstrates the necessary validity, number one, and number two, meets the operating environment requirements to be able to be used in clinical research. All of these teams have been influential in our work to date. Cloud UPGRS, a team out of London, and John Hopkins, I think everybody knows. And the Roche app that Florian's team did was maybe one of the first apps to be deployed in clinical studies, of sponsored studies rather than a research study. And this app, if you look at Roche's investor decks, has spawned a whole family of assessments into diseases like Huntington's and many others. However, I would like to single out, and I've left to last, the study on the left, the Empower study. What's unique about this study is that Sage Bionetworks put this study out there for the public to enroll in and use. But most importantly, they were generous enough to make their data accessible to researchers to understand and work with the sensor data and so on. That in and of itself is the single most enabling task for everything that we go through in the rest of these slides. Next slide, please. So let's dip into the UPGRS for a second. Aside from needing to go to a clinic to have the assessment administrated, aside from needing a trained clinician to do the work, there's some properties here and a picture on the right that kind of highlight the complexity and things you're going to have to deal with when you deploy a questionnaire like this. All the numbers in brackets refer to references that are provided in the last slide. So like any questionnaire, many questionnaires, we have to think about inter-rater variability. And for the UPGRS, this can be up to 20%. So in terms of 20% of a scale, you could make your own decision about whether that's a small or a big number, but it needs to be taken into account, as do all of the other properties on this list when you're defining your endpoints, you're defining effect size and sample size and so on. They're avoidable problems and you can work around them. We list them here because it's important to know that they must be dealt with. On the right-hand side, you have what's called a factor structure picture. I'm not sure if too many people will have seen a picture like this or not, but they're typically used to look at in an exploratory sense, to look at the stability of a questionnaire data set and look at the inter-item correlation. The way to think about this picture is to focus on the ovals down the left-hand side and then all the connecting arrows. What this picture is showing you is that each of the major factors that are the axes that define the variability within the data set are all interlinked. In clinical terms, it's not like someone has a gait and balance issue and nothing else, or that the severity of the gait and balance is measured in the UPDRS is completely distinct and independent of the severity of tremor. It's not. All of these items are interrelated. Next slide, please. So what do we do? Going back to the Empower data set, you get access to hundreds and hundreds of sensor data output. And you can take all that data and you can start to put it through either statistical analysis or signal processing techniques and so on. And in this example here, we walk you through a finger tap, which is a measure of hand dexterity, where the patient is required to tap the circles on the screen that are shown in the screenshot on the left. Each time they tap one of these circles, the position of the finger tap on the screen is being recorded. The timestamp of that tap is being recorded and so on. And you can see an example of the raw data in the middle picture. This is straight raw data that comes out of Apple's open source research kit platform, which we use for some things and not for others. You're also lastly getting the button that was tapped, be it with the left button or the right button. In and of itself, the raw data is useful. What turns out to be more useful is looking at the derived measures. Examples could be how many button taps do people get wrong? Did they go left, right, left, or did they go left, right, right, stop, left, left, or whatever it might be? And that's the y-axis on the charts on the right. On the bottom, you have time between taps, which is in seconds. And the distinction between the two graphs is that knowing that Empower was a supported public enrollment, on the left you have self-stated Parkinson's off state, and on the right you have self-stated healthy patients. When we first saw this picture, we were astounded because each circle in this picture represents one patient. And in the healthy subjects picture on the right, all the data is clustered together, which means that there's not that much variability between patients, and it's very difficult to distinguish one patient from the next. If you go to self-stated off-state on the left, you can see there's a much greater degree of variability in the data, and it's quite easy to pick out one subject from the next. So when you're thinking about the measures that you want to take forward to study a disease, this is just the kind of picture you need. Because if you can't tell a patient with disease from a healthy person, your measure's no good. And if you cannot tell the difference between one patient with a disease and another, you have very little chance of either measuring disease severity or disease progression. And so going through many different kinds of assessment raw data, this is the exercise that we went through to come up with a list of candidates from the Empower data. But that's only the beginning, and the remainder of the slides will take you through some of the other steps. But before we go down that route, let's have a look at all the kinds of things you can do here. You can look at different kinds of tremor, like kinetic tremor or postural tremor. Here you're interested in the amplitude and frequency of tremor. Finger tap I already covered. Speech, where you're looking at the perceptual coefficients, jitter, and fundamental frequency. Our platform also supports the ability to deploy assessments for cognitive impairment, pretty much any kind of questionnaire that can be confirmed through cognitive debriefing and usability testing that it's suitable for use. And then continuous measures from a wrist-worn device, actigraph or Apple Watch or whatever the customer's choice would be. What we're then wrapping around that is the ability to configure an assessment schedule. And some diseases like Parkinson's or others, the duration of standard of care medication and its effect on the patient is relatively short term. So what you really need to be able to do is to distinguish between different medication states, as well as being able to measure change in symptom severity over time. And to do that, you need a very dynamic schedule of assessments. It's not enough to be able to say, come into the clinic on a Monday or do your assessment on a Tuesday and hope that you captured the full spectrum of measures of interest and that they are truly representative of the general state of the patient's disease and severity. Next slide, please. So we've talked a lot about analyzing sensor data, but so far we've made the assumption that the sensors themselves are suitable and that it's in some way possible to analytically validate these measures. In the next few slides, we take you through some of that work. In the left-hand picture, you see a blue square, which is called a shaker table. And it's actually a table that was built for the civil engineering industry. What people use it for is putting models of buildings on top of it and then basically shaking the model to destruction. It's used a lot in earthquake tolerance engineering for skyscrapers and things like that. The cool thing about this table is that you can pre-program it to shake how you like. And so what we did is adapted this table with a cradle to bolt an iPhone and different devices to the top of it, and then to shake them through a full series of amplitudes and frequencies. And what you then do is compare the output of the device sensors with the calibrated output of the table. That's a foundation for checking whether the device sensors work over the range of frequencies and amplitudes that are required to fully capture the measures of interest. On the right hand, you see a summary of some of the analytical validation work that we've done, where you're asking a number of subjects to do multiple repeats of the same assessment. Walk for 30 seconds, walk for 10 seconds, walk for 5 seconds, rotate a phone, shake a phone, tap a phone, whatever it might be. And you're taking all that data and you're calculating the ICC of agreement between the scores observed by multiple raters for each test and the output of the algorithms. What we're aiming for here is as high a threshold of success as possible. And it should really be an ICC that's at least as good as the gold standard that you're trying to complement. Hopefully you can do better. On the next slide, please. So this is a slide which takes you through some of the sensor work, and at first value it's kind of complicated, but the conclusions are simple. On the right hand side you have a graph, and the y-axis is acceleration in g, just like you measure gravity from pretty much nothing at the bottom to around 3 and a bit g at the top. 3 and a bit g is quite a lot. When we were doing this testing, you move that shaker table at 3G and the whole office knows about it because it's loud and everything's vibrating. That's not accelerations that humans would perform, even in like an acrobatic stunt in an airplane. 3G is still a decent number. What you care about instead are the accelerations that are observed in the measures of interest like walking or tremor and so on. And acceleration in walking is typically around 0.5 G. The references providing these point estimates are in the back. Tremor, much, much lower, around about 0.05 G. And then across the top, you have three combinations of device and table: iPhone, Actigraph GT9X, and Actigraph Center Point Insight Watch. And all the colored dots are the ICCs of agreement between the device and table. And you see that in iPhones, ICCs are really consistently high. For the GT9X and CPIW, which are marketed for activity and sleep, they do very well at recording walking acceleration. What we learned for the Actigraph GT9X is that to capture accelerations in the ranges of tremor, you have to turn off sleep idle mode, which is a setting on the device, which we wouldn't have known if we hadn't done this testing. Next slide, please. Now let's take a dip into the algorithms themselves and the analytical validity from the test I described earlier. What you see here at the top is an iPhone assessment where someone's presses get started and then they walk, and then when the phone tells them to stop, they stop. A little bit similar to what was done in Empower, done by other people, but also with some differences. At the bottom, you have an Actigraph GT9X, which the participant is wearing for the duration of the study. Let's go through the tables now. What you see here on the top table is the ICCs for different base measures: duration, distance, steps, speed, and so on, for different kinds of tests down the left-hand side. The first row, we were really pleased with these results. The ICCs for most of the measures really pretty high. Stride period could be improved. We'll work on it. What we're also seeing here is variations. We can't expect a patient to always be wearing jeans or a tight pocket that would constrain the motion of the phone. So what we need to know is how well do the algorithms behave if people put the phone in a loose pocket, or if they don't have pockets and they want to put the phone in a bag and then do the assessment. What difference does it make? While there's a slight reduction, we were pleased to see that the results are pretty much comparable. And this gave us some good insight for the future. On the bottom table, you have a quite different table because what we're looking at here is our ability to detect walking in continuous accelerometry data from an actigraph. And now, instead of looking at the ICCs, we're looking at the mean absolute error, MAE, root mean square error, and mean absolute percentage error for many of the same measures. That when you look at measures of agreement put in these terms, you have to think about whether they physically matter or not. And we would suggest that a mean absolute error of less than one step is probably not something that we would worry about too much. But there are some bigger numbers here, like 3 steps or 2 steps. What we ended up here is realizing that if you look at the underlying raw data, that when people walk short distances, they don't really get into a full stride. And that really you need people to get into a full stride. Walk for a few seconds before you get reliable data. Next slide, please. So this is a sidebar, but it's one that we think is important to call out. When you live and work in this space, you get a lot of people telling you things are validated, and then they'll do things like quote you correlations or R-squareds or whatever it might be. There's some very different fundamental differences between all of these measures, and we're planning to do a white paper that takes you through a lot of this in more detail. But the important point to note here is that correlation and agreement are not the same thing. Correlation is an indicator of a relationship between two measures, but it's not a measurement of agreement because it doesn't require the measures to be measuring the same concept or same underlying concept. And they could be biased. They need not be related at all. They just happen to show a correlation. Intraclass correlation coefficients, on the other end, definitively has a single measure of agreement that takes into account both correlation and bias. And in our early algorithm development work, we were seeing prototype results that had good correlations but poor ICCs, and it was a abject lesson to us about how to think about measuring agreement and what you need to do to be able to improve agreement and work accordingly. That said, they each have different purposes and different limitations, and it's not to say that any one particular statistic is good or bad. It's more that you should call a statistic what it is and use it for its benefits and none of its limitations. Next slide, please. Let's dip into continuous gait and balance for a minute. What you're really trying to do here is pick out when people are walking, because you don't want to count steps when someone's asleep. You don't want to count steps when someone's sitting in a chair. You only want to count steps when they're actually walking. And so what we came up with is a method to be able to figure out in raw accelerometry data when people are walking. And you see a sample trace from some of the validation work that we did at the bottom. In this case, it's pretty easy to see when a person's walking because they walk and then they stop for a defined time, and then they walk and then they stop for a defined time. And what you're looking at here, back to the mean absolute error measures that we were seeing in the table a couple of slides ago, is how accurately does the walking detection algorithm pick out that someone's walking versus the actual absolute time in the trace on the bottom when they did actually start and stop walking. Once you have walking periods, you can apply your gait measures algorithm to each of those walking periods. So what we have now is the ability to calculate gait measures in a both wear independent way and a device agnostic way, both at a point in time because you're telling someone via an iPhone to walk, or in continuous data when you're having to detect whether they're walking or not. They both have a place. A point in time assessment, I like to think of it as a as the ability to generate a signature, because it gives you a very clear picture in a relatively constrained setting of how somebody moves. But that's not the same as activities of daily living, and to some extent, a point-in-time assessment of gait and balance or any other objective measure has many of the same limitations as a point-in-time assessment on the questionnaire. It's a time point. You don't know if you captured a patient on a good day or a bad day, or whatever. They might have a cold or a cough or otherwise feel under the weather, and you don't know. So capturing the same measures within the context of activities of daily living is, we hope, an important step forward in the overall field of research in CNS disorders. Next slide, please. So some of the algorithm work we've, in a pandemic world, been limited to publishing in posters at the present time, mainly to get feedback. When you live and do this work every day, you don't really know how good or bad it is, and you don't learn from the industry. And so in the last few months, we've had the opportunity to put some of these articles out there, which speak at a high level as to the work we're doing and what we're trying to achieve. Next slide, please. That's now over to Chris to add content that only he can, and I probably never will be able to. So,
not at all, Rob. Thank you. I'm sure many of you can see from Rob's presentation why his fundamental training as a physicist is really, really valuable to how we think about solving some of these problems around measuring movement and doing it in very precise and methodical ways. One of my favorite quotes when I got into this field, I'm going to attribute it to Barry Peterson. Barry was a longtime Pfizer researcher who subsequently was medical director of Philips Respironics, which is a great device manufacturer with whom we've worked. He's a consultant now. And Barry liked to say, medicine is defined by what we know how to measure. I don't know if Barry originated that quote. I think maybe he did. It's an incredibly smart one. And it's insightful into some of the significant limitations associated with scales that we use in things like drug development. As many of you know, in diseases like multiple sclerosis, migraine, Batten's, as is referenced here, we deploy composite scales that look at the multiple symptoms, similar to the examples Rob showed you with the UPDRS for Parkinson's. And yet, in fact, the realities of disease as we continue to understand disease subtypes, be they genetically defined or occurring for other reasons, the reality is some patients experience an abundance of one symptom domain and not another. A multiple sclerosis patient might tell you, and of course, talking to patients is always one of the most important ways to understand and improve upon these scales, that the challenges with their manual dexterity mean that it's hard to open a water bottle like I'm holding right now and unscrew it, or a jar of food, but they might have less difficulty walking. You might find that a patient with Parkinson's has challenges with speech, but not with other aspects of tremor, and so on and so forth. And so in many of these diseases, one of the challenges, particularly around developing new medicines, is that these composite measures that we have don't necessarily really reflect where patients might see improvement on one domain. Their absolute score may not move in a meaningful way because other domains are not only unchanged, but perhaps not even important to that particular patient. So we're always looking at ways to better deconstruct and appreciate what are the measures that matter to patients, and are there ways in which we can build more effective scales or instruments that capture those changes. The other feature of clinical development scales or performance assessments that I'm sure many in the audience are familiar with are the use of these performance assessments like the timed 20-foot walk test or the 6-minute walk test. The simple way I think about this is they're designed to remove human error and bias. It's not that easy to look at someone, even if you're a trained neurologist, and quantify how they're walking. But if you have them walk for a pre-specified distance under carefully controlled conditions, it hopefully allows you to standardize that measure to some extent and make comparisons across patients or track disease progression within a patient. The challenge, as many of you know, is that even with that level of standardization, we still see a lot of variability when applying these measures in different disease areas. And finally, it's all too often that we take tests that we know how to do. And the 6-minute walk test is a classic and commonly picked on example where it's validated to predict outcomes in diseases like congestive heart failure or COPD, but we then choose to take that test and apply it in other conditions where it may or may not really be relevant to what patients care about or ultimate disease outcomes. So these are known challenges and limitations of these kinds of scales. In each of these, we are actively working on or have worked on digital measures that have opportunities to improve. Aspects of walking speed have been shown to be incredibly predictive of disease outcomes in a lot of different disease areas. And in fact, as I think Rob did a great job demonstrating, we have technology that can do a phenomenal job of detecting and characterizing walking speed without necessarily having to put someone through a prescribed exercise like one of these tests. Although what we have also developed in the case of things like 6-minute walk test is the ability to guide patients through an assessment using technology on a phone so that we can create apples to apples comparisons in the cases where you want to understand how a digital measure directly relates to traditional data that we might already have, such as 6-minute walk test values and so forth. So we see and are working on opportunities in all of these areas, figuring out how to digitize something like the 25-minute walk test, getting that done effectively alongside activities of daily living for multiple sclerosis could be highly impactful. In migraine, one of the challenges around migraine therapies is understanding the prodromal symptoms and knowing when a headache is going to come on, and then understanding what is the implication for that patient's ability to function, move around, and do things that are important to her or him. Diseases like Batten's, and that's really just one example of many rare diseases that have disability scales. The PCAN DRS is also referenced here, have a great deal of movement disorders that are similar to some of these other diseases and are natural opportunities where these modules can be applied and are being validated to determine whether or not these diseases behave in ways that can be characterized similar to other diseases in which we've already generated data. And finally, something like the 6-minute walk test, again, as frustrating as it may be to many researchers, it is widely used in so many conditions. And there is so much existing data on it that we felt compelled to figure out how can we do this digitally in such a way that it can be used as a companion tool, and again, creates the opportunity to assess patients at home and more frequently than you could, especially in a COVID environment where access to clinics, particularly for sick patients, is something that we want to heavily limit. The next slide, please, Adam. So some key takeaways. The generation of natural history data, registry data, and open source studies is incredibly valuable to anyone developing digital measures. I was fortunate to work closely with Sage BioNetwork's founder, Stephen Friend, and have had great collaborations with people like Lara Mangravide, Larson Omberg. There's too many great people there to possibly mention and give shout outs to, but the whole team behind Empower and at Sage BioNetworks was instrumental in creating the first proof of concept for what could really be done on a very broad scale, collecting data from patients, in this case, in the Empower study with Parkinson's data. And the utility of that data is just incredible in terms of learning things about user behavior and how to model some of the tools that any of us may want to build and improve upon in order to create disease assessments for different contexts of use, such as drug development. Research Kit, Apple's Research Kit is an incredibly important and useful tool that has helped us advance our work. But it is important to understand the complexities of the data model, and that in many cases, we are essentially getting a relatively raw canvas, a tool that can be used to collect data or a basic framework for doing so. But the actual validation and application of that data is typically work that needs to be done by those of us who want to build and apply tools in this space. So we're very grateful to Apple for providing it, but also conscious to make sure that we fully articulate what it provides and what it does not provide. As Rob touched upon, I think it's so critical that when we're evaluating the claims of different tools have been applied. And particularly for those of us that work in drug development, recognizing that the things people may be assessing for, if you're looking at care management or just modeling disease progression, are often not the same questions and don't have the same statistical tests applied to them. So while correlation is often relevant, correlation without agreement or with poor agreement can be problematic. So understanding that empirical measures should also agree, such as distance and speed, and understanding that appropriately is critical to figuring out how these tools can be used effectively to do things like identify the treatment effect of novel therapeutics. Finally, as I was alluding to on the prior slide, a lot of the instruments that are currently outcome measures that are accepted for drug development and other uses are broad composite measures. Our initial instinct was very often to want to create like for like assessments. So for example, if you're doing a digital 6-minute walk test, how does it compare to the in-clinic test? If you're doing a test of tremor with an iPhone, how does that compare to the way tremor is assessed in the clinic? Those are important questions to consider. But broadly speaking, a digital set of tools is not going to do the same thing as the in-clinic tools. And quite frankly, what we've realized talking to clinicians and talking to patients is we don't want to do that. We don't want to replicate some of the inefficiencies or problematic ways in which we're assessing things because we're limited without technology today. We want to take advantage of the potential of technology to build better assessments. What that really requires is that we look more at the frameworks for validating patient-reported outcomes and creating new outcome measures. So it requires both qualitative and quantitative input from patients. And it really, I would say methodologically, ends up mirroring more how you validate and develop a novel PRO, even though it might be a technology or performance assessment, than simply doing a like-for-like comparison of two assays. So I hope you've all found this talk helpful, and we've tried to allow 10 minutes for questions. So I'm going to pause here and take a look at the Q&A from my colleague Adam, who is moderating.
There's one question. In your experience, what is the best way to communicate the value add to digitizing endpoints to our colleagues? I think we can both speak there. So,
Rob, I'll take that one. And if you look over at the panelist chat, you'll see some other questions that are just for you. So you look at those while I start here. I think, so the value add for digitizing endpoints, I think it's incredibly important, not to oversell and to be realistic about the progress and investment that's required to develop new digital endpoints. But I would break it down as simply as follows. First of all, digital endpoints have the opportunity to more objectively assess symptoms of patients in the place and time that matters to them the most. No one cares how far that they can walk inside a hospital clinic in 6 minutes. I think no one cares about that. They do care about things like, can I get up and move around the house? Can I go to the grocery store and walk independently? So whenever we figure out how to move measures closer to where patients care about them, and then also characterize them more objectively than we realistically could through an in-clinic setting, that is a huge benefit for everybody involved in the process. The second thing I would say for those of us involved in drug development, and I assume that's a lot of the audience here, the opportunity to create more sensitive measures can be incredibly important in figuring out how a drug is working and on what symptom domains. And very often with a smaller number of patients or in a shorter amount of time than if you're limited by very infrequent assessments that occur in the clinic. Of course, there's nothing that we can do that changes the actual progression of disease. We can't necessarily always observe it faster than it actually happens. So there will always be intrinsic limitations, but the opportunity to do more targeted, more frequent assessments is incredibly important to the productivity of drug discovery. So those would be the key selling points from my perspective. Rob, have you seen the question in the other chat about?
Yeah, I did see the other one. I see one question, two, maybe 3 questions. All right. How much evidence does a regulator need before they will accept a digital endpoint? The somewhat simple answer is an awful lot. If we had known at the beginning of this journey how many pages of documentation, I think you would end up contributing to. If um, I wouldn't have said the right number. Um, the 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 decision points really, I think here I'll say a couple of things. A lot of times we'll go and meet with sponsors and they'll want to think about something relatively simple and exploratory as a test and learn, but they'll have an eye to the future. And future there means at least a secondary, if not possibly a primary and a phase 3. The I would turn the advice around another way and say, if you have any intentions to go beyond experimentation, then the sooner you start to consider all of your work as a journey towards a regulatory filing, the better. Next question. You mentioned a wrist-worn device. Can you do this work with an Apple Watch? We Actigraph have been extremely good partners of ours for a long time, and they have a very good system and good platform that you can know and trust with a good degree of confidence. So we get asked questions about Apple Watch all day, every day. We have done some research work here. There are definitely some advantages. Of course, it's a cool device, but I still think that there's a good amount of work to be done to validate the operating environment and the data processing that comes out of an Apple Watch. We've certainly looked at this in a lot of detail for gait and balance, especially, where I think the picture is pretty good. I think one day there might be more choices on the table. The advantage of an Apple Watch is that you can get multiple different kinds of measures out of one device: heart rate, gyroscope, accelerometer, and so on. The extent to which you care about their validity really depends upon the algorithms that you use to collect the measure as provided by Apple, or the algorithms that you use to process the data. That would be my view there. The other thing I would mention is that some of the most significant qualities of clinical grade devices, like an Actigraph, is they can run on the same battery for days and days and days and days. If you have a patient in the study, one of your first concerns is to remove as much of the operational burden as possible. No matter how cool an Apple Watch is, I think it will be a long time before an Apple Watch has a battery that can last for the same kind of timescales. And so thinking about the operational burden of an Apple Watch is as important as anything else.
Thanks, Rob. I want to thank our attendees, and of course you, Rob, for doing the heavy lift of this presentation. Thank you for everyone that joined and submitted your questions. We will be posting an archive of this webcast on